My friends, we're going to keep this rolling. I get to bring back on a fan favorite, a good friend of ours, Mr. Luke Xavier Jacoby. Is Xavier your middle name? It might be. I don't know.
It's not. I wish it was.
It
It's a lot more fun
sounds
than
kind of cool.
Maybe easier to spell, maybe not. I may or may not have been denied a driver's license when I was 16 because I spelled Benjamin wrong.
Oh,
But
boy.
anyway, we'll put that all behind us and keep plugging forward with this panel.
All right. I'll let you get to it.
Awesome. All right, everybody. I'm very excited to be bringing us our very first panel of the day. Title of this one, Cancer Immunotherapies, Fighting Cancer with the Power of the Immune System. We've got an awesome group here ahead of us, and we're going to be getting all of the stock tickers in the chat. So if I talk too fast or not clearly enough, don't worry. Look at the chat. You'll have them there. But staying on the stream with us, Dr. Frank Beto-Otto, CEO and Director at PDS Biotechnology, Dr. Mark Newman, Chief Scientific Officer at GeoVax, and Snehal Patel, CEO at Greenwich Life Sciences. Hello, everybody. How are you doing today?
Good. How are you?
How are you?
Doing well. I don't know how much of my background you can see, but we've got this nice Detroit spring coming in. It's finally out of the snow. It was like a very dreary background for quite a while. So all is good in my world. And hopping into it, everyone, I think it would be helpful if you could just do an introduction of yourself, the company, and how far along you are, and let's go in order. But Frank, you want to kick us off with that one?
Sure. So PDS Biotechnology is a mid-clinical stage cancer immunotherapy company, and we have 3 phase 2 clinical trials ongoing based on our company's proprietary Versamune technology platform. Versamune is designed and developed to prime the immune system to effectively recognize cancer, generate the tumor-attacking killer T cells to effectively attack and help eradicate the cancer. And so that's really the purpose of the Versamune technology and our key focus. We're also applying that technology to developing a novel T cell activating COVID-19 vaccine. So that's also being done in parallel with our oncology programs.
Excellent. And Snehal, I'll pass the same question over to you. Just a brief introduction of yourself, the company, and how far along you guys are.
Sure. My name is Snehal Patel. I'm the CEO of Greenwich Life Sciences. So we're working on a 9 amino acid peptide called GP2. We're focused in breast cancer. So we are treating patients after surgery, after they get a year of Herceptin or Kadcyla treatment, and then they get our treatment, which are 11 vaccinations over 3 years. So we've reported 5-year data from a phase 2B trial in December. So we've shown that we've seen no breast cancer recurrences over 5 years. We have no serious adverse events. Product is very safe. So that data was reported in December. We'll have some immune response data reporting in April. And we're planning a phase 3 trial that we hope to start in 2021. Thank you.
Excellent. And Mark, same question over to you. Just introduction, where you guys are, et cetera.
Okay, I'm Mark Newman. I'm the Chief Scientific Officer of GeoVax. We're a clinical stage company, but we have been focusing on infectious diseases that are just now venturing into the cancer immunotherapy world. We have phase 2 products for HIV for prevention and phase 1, and are involved in phase 1 testing for therapy, the HIV cure programs. Our technology is based on modified vaccinia virus ANCRA as a viral vector. And the goal is, as you've heard from the others, is to induce T cell responses primarily to control or eradicate viral infections. And now, as I said, moving into cancer.
Excellent. And one of the things that I think is really interesting about this panel that we have here is that you're all taking different approaches, right? We're somewhat working towards the same end, but with different approaches. And so the question that I want to ask, is one, this is sort of a bundle question, but one is, what are the hallmarks of an effective treatment? And then can you dig into the approach you're taking, why you're taking it, what makes it compelling, et cetera? And Mark, if you could keep us going with that one, again, hallmarks of an effective treatment, and then dig into the approach a little bit for us, please.
Okay, sure. So I think what you've already heard is that we're focused as a group on inducing T cell responses. These are well known as being effective at controlling, viral infections, but also in tumor immunogenicity. What we're working on at GeoVax is a little bit different. We're focusing on altered mucin, or the MUC1 gene. This is a well-established target for both antibody and cellular immune responses based on epidemiology studies. So our technology is designed to induce both an antibody and a cellular response. Because it's a viral vector, you're getting the cellular responses induced because you get in vivo production of the transgene, in vivo production of the vaccine immunogen. But our technology allows us to express these virus-like particles, which cluster the altered mucin gene on the surface of a virus. Looks like a virus. It's a particle in the body, and this gives you the multi-dimensional structure that is critical to inducing high-affinity antibody responses. So it's kind of a two-step approach.
Okay, excellent. And Snehal, same question over to you. What are some of these hallmarks of an effective treatment? You guys have clearly put out some astounding data, and then the approach. If you could dig in there, I think that would be helpful for the group.
Sure. We're doing something really simple. We're injecting the 9 amino acid peptide in between your skin layers, and we're getting a CD8 T cell response. We can measure that immune response by testing to see what the injection site reaction diameter is. And we can give the drug without anything else as a separate test. And the patient comes back 2 days later and we measure the diameter of that immune response. We can measure the T cells by taking a blood sample and quantify the number of T cells that respond to the peptide. And we can also do an ELISpot assay to measure the functionality of the T cells to see if they're functionally performing. And all three of these can be done for many different technologies. So immune response is very important, but you also have to have clinical outcome, because you may be targeting something that's irrelevant. And so you may see a great immune response, but you may see no clinical outcome. So to me, those are the most important things you look for in immune response, but you have to wait for the clinical outcome too. You can't count on efficacy if you just get an immune response, my personal opinion. And then also safety, right? So you want some injection site reactions, you want some response to the injection, but you also need it to be tolerable. So you need the balance of that as well.
Yep. And Snehal, I just want to follow up on something you said about the difference between the immune response and then the clinical outcome. Can you just, for folks who might not be aware, could you clarify the differences between those two?
Sure. I could train your immune system to attack some foreign object and get a great immune response when I trick the immune system to attack that foreign object. But if that foreign object is not the target that you're trying to eliminate, so for example, if you're tricking the immune system to attack some part of the cancer cell, but that part of the cancer cell is not being presented by the cancer, then you're probably not going to see a clinical outcome, even though you get an immune response.
Okay, excellent. Thank you.
Mm-hmm.
And then Frank, same question over to you. Digging into the approach you guys are taking, why you think this approach is compelling, and then some of those hallmarks of an effective treatment.
No, I think so from our perspective, we've looked at it from what we know about immune oncology today and how tumors behave. So we know that tumors are able to survive in the body and grow because they actually co-opt a number of our body's regulatory mechanisms to essentially hide from the immune system, right? So they hide from that immune system surveillance. So one of the key things we believe is very important is to be able to expose the tumor and make it much more visible to the immune system, right? So take, for example, if you decamouflage an enemy and make that enemy more visible, that enemy doesn't get attacked until you have the soldiers and the ammunition to go attack that exposed enemy. So exposing the tumor is very important, but also generating the right kind of T cell that can then see that exposed tumor, go attack and kill those exposed cells also becomes extremely important. one of the biggest limitations in immunotherapy today has been the ability to generate those tumor attacking killer T cells in vivo in patients, right? So what our technology does is the Versamune technology has overcome that obstacle of being able to take that protein that is unique to the tumor. So to train the T cells to recognize the tumor specifically, we take a unique protein from the tumor, combine it with our Versamune. Versamune is then able to present it into the right compartments of the immune system to train our killer T cells to recognize that target as a foreign agent. Now that's one step. Training the T cells is one thing, but providing them with the ammunition is a whole different step. So it also activates what's known as the type 1 interferon signaling pathway. That's critical to provide the right immunological chemicals to help these T cells to proliferate, expand, and enhance their killing potency. So by doing those things together, making sure that all those pieces come together effectively, you can then develop an effective immunotherapy that can go in and effectively kill the cancer cells. Now, that's one reason why we've started our initial clinical trials in what you realize combination immunotherapies, right? Because we're saying, okay, the immune system, multiple arms of the immune system have to be activated, and the tumor is co-opting multiple mechanisms to hide from the immune system. So for example, in our combination with Keytruda, for example, Keytruda blocks what we call the immune checkpoints, which is one means by which the tumors hide. So blocking that and exposing the tumor is what Keytruda is doing. We also do that by a second mechanism. So now we have two mechanisms to expose the tumor, and then we generate those powerful killing T cells, which can then come in and kill that exposed tumor. And so that's the reason why we are starting our clinical trials with the combination therapies, to maximize that potential to expose the tumor and have the immune system more effectively attack and kill those exposed tumors.
Yep. And I was going to say, Frank, you're giving me a great segue, because the next thing that I wanted to talk about was combinations, partnerships, et cetera. You know, from the investor angle alone, you know, it's absolutely interesting. And from the scientific angle, you know, when we were preparing for this, you guys definitely talked about some of the benefits there. You know, so if you could dig a little bit deeper into, you know, the combination approaches you're taking now, how partnerships may play a role in the future, et cetera, that'd be great.
Sure. So I think partnerships are going to be, especially combination immuno-oncology, I think is going to be the next frontier in immunotherapy, and how a lot of these cancers will be addressed using immunotherapy, simply because of the multiple mechanisms that have to be overcome and also addressed. So for example, taking the National Cancer Institute led trial that we're talking about, these trials are in patients who have actually failed initial treatment. So these are really sick patients. And so we're looking at a combination with, this is actually a little bit different because it's a triple combination. So we have 3 immunotherapies combined and being given to these patients. One is what we call a bifunctional checkpoint inhibitor. So it exposes the tumors and also attacks the tumors by a specific mechanism. The second is what we call an immunocytokine. It's targeted to the tumor, so it actually enhances the T cell responses within the tumor itself, right? And then we have PDS-0101, which is trained to generate those T cells to attack the tumor. So the National Cancer Institute actually published a study with this triple combination where they showed that with PDS-0101, we generated a much larger number of killer T cells, but very interestingly, we had very few clones, because now the T cells, even though we had a much larger number, we're now trained to specifically recognize and attack the cancer cells. And we saw a significantly enhanced anti-tumor effect. That's now being studied in patients in a phase 2 trial. And I mentioned earlier that the highest efficacy rate in this specific patient population who've already failed cancer treatment was about 30%. And so what we said was that, look, even in these very sick patients, if this triple combination is actually working, we would want to see probably at least a 30% improvement over the current standard. And so with the first 8 patients, if we were able to meet that 40% threshold, showing that we are actually able to generate these T cells, very likely in these very sick patients, we will continue the trial. And that's what we demonstrate, that when we were able to see those at least 3, even before we reached the 8 patient threshold, which was a very good sign that it seems to be very likely that what we saw in the preclinical models is actually translating to humans. And so we've now opened up the trial to 45 patients to perform the full trial to get a much better understanding of what's happening. But that's our approach. So all three of our initial trials are in combinations, different combinations, to really understand how the Versamune technology becomes complementary to some of these current treatments. And we see that as probably the least risky and the most rapid approach to commercialization, because if we can go in and show that the combination is superior to the current standard of care, we believe that provides us with a very clear path to commercializing the product. And safety also becomes very important, right? So we want to improve the therapeutic benefit to the patients, but we don't want to compound the toxicity either, right? So both components become very important in evaluating these combinations. And so far, the PDS, the Versamune technology, has been very well tolerated by the patients. So we are quite optimistic about the potential of these combinations.
Excellent. And Mark, I want to ask you the same question. You know, over at GeoVax, how are you guys thinking about combinations, partnerships, et cetera?
Well, there's a two-part answer here. The first part is the simple one, and that we view the vaccines that we will produce as being evaluated and used in conjunction with standard of care. So our assumption is that we will probably be piggybacking onto the checkpoint inhibitor types of programs. That this is what we've heard from our discussions with the NCI. The combinations of the products a little bit different. As you're hearing, the goal is to train and expand the correct T cell population. Using the viral vector, it basically has self-contained adjuvant properties. So it's a single component, but because it is a virus, it induces the type 1 interferon pathways, the early cytokines, the chemokines, the internal pro-inflammatory responses. All of these things are induced as a function of the vector. And then we're also looking at combinations to narrow down or focus the immune response by doing what we call heterologous prime boost. So that is to immunize with, for example, a synthetic peptide. You're hearing about a 9-mer. We would immunize with the 9-mer and then follow it up with the MVA to expand the population, or do the alternative, and that is to immunize in a combination with the MVA first to intersect that intracellular pathway, induce cellular immune responses, and then augment it with a peptide booster. So these are all open, and they're all part of the program we are developing. As far as commercial partners, we have not gotten to that step yet. As I said, we've kind of pioneered this with infectious diseases and are now stepping into the cancer immunotherapy programs.
Excellent. And Snehal, you mentioned some very, very good data that you guys put out earlier in this panel. Was that part of a combination treatment? If not, how are you guys thinking about combinations and partnerships, et cetera?
Sure. So yeah, GP2 is really from the beginning a combination. Our theory from the original investigators is that you have to have a healthy immune system. So you can't be destroying the immune system with chemo or radiation and then expect the immune system to respond to your therapy. So that was one of the first things we had to find the window when we could treat them. Unfortunately, one of the first treatments, and you can call it a combination, is to have the surgery so that you can reduce the burden and help the body heal so that it could be prepared for a treatment like ours. The second product is an antibody called Herceptin, and that antibody helped to reduce the recurrence rate in half. And then our treatment started after Herceptin, and we reduced the other half of the patients that were recurring to 0. So that it's really important to think about, if you're going to do combination, those products need to be approved. As Mark mentioned, they're called standard of care because then you can use them. But if you need to use an innovative product in combination, then you have to work with that company. That company has to be willing to work with you. And that's a collaboration, it's not a partnership. But those are the things that I think are going to be more important now, especially, for example, in those areas where nothing is effective alone. These companies have to come together and put their products together. They're both unique, neither is approved. and they have to figure out how to work together.
And that's the collaborations that you're saying are you expect to be a bigger part of the future, correct?
Yes, yes. So for us, we're going to need to collaborate with companies that have better versions of Herceptin, that have other peptides. Everybody who wants to work with Merck has to collaborate with Merck to get their checkpoint inhibitors. These are agreements that have to be made, but Merck doesn't necessarily have marketing rights to your product. So the second part of your question is the partnership, and that partnership is for the marketing rights to your product for a particular country or globally. And that's a completely independent financial decision that you can make. In our case, we're funded through the end of the interim analysis and hopefully the filing of our BLA, so we don't need to do something soon, and that gives us more leverage. Other people do it sooner as a way to fund the trial, and then they have to raise less capital. So there's two ways to do that.
Excellent. And we just have a couple minutes left here. You know, something that's definitely important for our investors out there. I see some questions in the chat. Is what's up next? So if you could talk about, you know, what next piece of data we'd expect to see, or announcement, or anything along those lines, and then also, you know, how we can stay in touch. You know, track the progress. That would be amazing. Snehal, could you answer that one first, please?
Sure. The American Association of Cancer Research has a conference starting in a few weeks from now. So on April 9th, they'll announce the abstracts. April 10th, they'll publish the posters. So we have a poster about the immune response coming out, which we've announced is coming out. We've not published any information about it. So this will be one of the missing pieces. It'll explain if we're seeing a immune response in all the people that we treated, irrespective of you know, which type of breast cancer they have. They could have HER2 1, 2, 3, 1 plus 2 plus or 3 plus stages of types of cancer. So it's really you know, important to have not only the final outcome of the trial and the clinical outcome, but also the immune response. And then eventually we'll be announcing the safety, hopefully sometime later this year as well. And these will be the final 5-year analysis for our phase 2 trial.
Excellent. So we've got a couple things on the radar, and that first one you mentioned was April 9th, correct?
Correct. That's correct.
Awesome. All right, Mark, same question over to you. What's next? What should we be on the lookout for?
Well, as I mentioned, our program is being transitioned from the infectious disease world. So we are actually in animal model testing right now, and discussions with NCI and trying to work out what we believe will be the optimal combination to be moving into phase 1 trials. You know, our goal is to be entering the phase 1, which would be, you know, dose escalation, safety, toxicity type of trial early next year, Q1 to Q2. So the first clinical event is not going to be until 2022.
Okay, excellent. And then Frank, could you round us out to tell us, you know, what we have to look forward to?
So as I mentioned, we have 3 phase 2 clinical trials currently ongoing. I talked about the preliminary data from the National Cancer Institute-led trial, where we saw at least a 30% improvement over the current standard. We expect to have more mature data on that program, hopefully sometime next quarter. There's a potential that we will present these at some of the leading oncology conferences coming up, but we'll typically make the markets aware of this when the data comes out, when we're going to be presenting at conferences. With the combination trial with Keytruda, which is the partnership with Merck, we anticipate that we will start reporting data from that trial probably in the 4th quarter of this year, or probably the 1st quarter of next year. And then the third one is the MD Anderson-led trial in cervical cancer. Again, the timeline for this is very similar to the Merck trial, where we anticipate that we would start reporting data in the 4th quarter of this year or early next year. For the COVID-19 program, we're anticipating initiating clinical trials probably late next quarter. So the nearest term would be the NCI-led triple combination. We expect data from that trial next quarter, and also hopefully to initiate the COVID-19 trial late next quarter also.
Excellent. So just to recap, everyone, Snehal, he mentioned April 9th and then additional data out later this year. Mark starting 2022 as a very big year for them. And then Frank, in the 2nd quarter, this upcoming quarter that we have, is when we'll get additional data from them. Excellent, guys. Thank you so much for joining us. It's an honor to have you here. I mean, I feel so inadequate on this panel, just here talking about stocks all day while you guys are out, curing the world's problems. So thank you so much for joining us.
Thank you.
Thank you very much for having us. Thanks a lot.
Absolutely.
Great job. Great job, everyone. Thank you so much for that. Again, you guys in the chat, tickers there are GLSI, PDSB, and GOVX. And Luke, you are not inadequate. Stop it.
Thanks, Patrick. I appreciate it.
I am here for you. Here for you. Really, really cool stuff, though. It's interesting seeing how some of this is unfolding, you guys. So thanks. Keep it up.